I was giving a presentation recently about my research project, and when I showed this slide, which is about the differentiation process of the malaria parasite within the mosquito when the parasite invades the mosquito midgut. And one of the researchers didn't quite understand what was happening, what I was trying to say. And that was because my drawings. She said that these drawings were not conveying the information that I was telling them about. So she is right. I mean, these drawings are not accurate and they may not represent what I was trying to say at that moment. So this is when biological illustration is really important. And why it is important? Well, first because an image, a picture, tells more than 1,000 words, right? And also because it is a universal language. Anyone can understand them. And if you search something on the internet, I guarantee you that there are illustrations, images about them, about that topic. Guaranteed. There are so many illustrations, and that is because they convey the information in a very efficient, fast, and accurate way. So scientific illustration, it's a topic on its own. There are many really good illustrations out there. For example, this is one of my favorite books, the Brusca of invertebrates. And look at these drawings. These drawings are beautiful. They are masterpieces. Okay, maybe my camera isn't good enough, so let's check it on the internet, a digital version. And as you can see, these drawings are extremely beautiful. Look at that. However, it is not the beauty of the drawings itself that it's important. Actually, for example, we have Nether. This is a very prominent and it was a very prominent and important medical illustrator. And his drawings are not really the best ones. They are not the most beautiful ones, but they have something. They have something that really helps you learn the topic you are studying. For example, and he even said it. As he wrote in 1949, clarification of a subject is the aim goal of illustration. No matter how beautifully painted, how delicately and subtly rendered a subject might be, it is of little value as a medical illustration if it does not serve to make clear some medical point. So in my case, my images were not serving to make clear the concepts I wanted to explain. So biological illustration has to be, yes, it has to be accurate, but it doesn't need to be perfect. It needs to serve the purpose of teaching, okay? And if you think about it, making pictures, making illustration, paintings, and getting to know nature by drawing and teaching about nature through drawings dates back even written language. Just after oral communication, it goes painting. For example, we have these paintings that date back to 30,000 years old, and look at this beauty. This is a study of movement. Look at these rhinos. I mean, these are studies about nature, about animal behavior, about animal anatomy, and it is an excellent way to teach others about these things. And I like a lot this quote. Once Picasso was visiting one of these caves, and after looking at them and studying them, he said, "We have invented nothing. We have learned nothing in 12,000 years." And I mean, yeah, people have been drawing way back before writing. And actually, some writing systems, for example, kanji in Chinese and Japanese Chinese language, are basically drawings, stylized, standardized drawings of stuff, of nature, of things, of ideas. And we have this also in Egyptian hieroglyphs, other logographic languages or pictorial languages, pictorial writing systems like in Mesoamerica. So yeah, illustration, drawing, painting, nature, conveying information, transmitting information through drawings is really important. And in the case for scientific illustration, biological illustration is super important to make the images, to make the drawings easy to understand and to clarify the topic you are telling about people, okay? And particularly in biological illustration, it's in biological drawings is really important because even some species, so the holotype of a species, the reference of a species, sometimes is a drawing. For example, in the case of plants or protozoa, they are really hard to preserve in a museum. So most of the descriptions of these creatures are through illustrations, and they serve as the true holotype of a species, right? So, just for example, these are some drawings I made when I was studying, and I'm showing them not because they are beautiful or because they because they are good drawings. Actually, I are really bad at drawing, but I wanted to show you because this is how I learned. This is how I learned for 5, 10 years. And these drawings are almost 20, more than 20 years old, and they are still valid. I can restudy them. These, for example, are some drawings I in my lab logbook that I make some observations of larvae, the materials, and how to prepare them. I was studying the eggs and how the mosquito larva hatched. Some animals and protozoa that lived within the. So yeah, biological illustration is really a central part in learning. For example, these are some drawings of the different forms the ookinetes of Plasmodium can acquire during its traversal through mosquito, and I even found the drawings, the first drawings I made about the differentiation process that I showed you in the beginning. These are totally useless drawings because they are not accurate. Um, they are an oversimplification, which is not good. And but one question may rise here, and it's why no photos? Why don't you take just photos and show the photos? Well, the thing is that photographs show the specimen as it is, and sometimes it can be difficult to interpret what is happening. For example, I have been looking at these parasites for hundreds of hours, but someone that hasn't looked to a parasite before might not understand what it's seeing. I mean, these are complex images. There's a lot of stuff to see here. Also, biological drawings can highlight some important features that you want to portray, or the hide other stuff that is not important for your specific needs at the moment in communication. So, how do you make biological illustrations? One way to do it is uh, by hand, just having a specimen, looking at it, taking some measurements, and just start drawing. Now, this requires a lot of skill, a skill, for example, that I don't have. I really don't know how to draw. So another way, and this is how I drew these nematode illustrations. These are very standardized and accurate drawings of nematodes. These are nematodes of bats that I drew quite a while ago. And I made this with something called a camera lucida, which is, it's also called a drawing attachment to the microscope, where you can see your hand, the pencil, and the paper you're drawing on inside of the oculars of the microscope. So this attachment projects an image of your hand and the pencil into the microscope, so you can kind of just trace the specimen while you are seeing it. And this way you can get very accurate drawings. Even you can see live, you can draw live specimens if you want, if they do not move quickly. But I do not have a camera lucida at the moment, so another way is to take a picture, print that picture out, and then place it on top of a light box, which is simply a box with a light source inside, that when you turn on, it shines the light through this acrylic or piece of glass, where you put on top your picture, and on top of that you place a paper and you just trace it. But another problem is that I don't have a light box. Thankfully, they are really easy to make, so we're going to make one. In this case, what I did was to just grab a cardboard box, cut the lid open, just a big opening. Then I also make an opening in one of the sides to get the cable of the lamp through there. And just place the lamp, place a piece of glass on top. It would have been better if this piece of glass is translucent. That way it will diffuse the light a little bit more, but it's not a problem. If the light is too intense, you just place some papers below, and that's it. For the drawings, I'm using stylographs of different nib widths, and also mechanical pencil and some erasers. So first, I'm going to trace the parasites with pencil, and then I'm going to draw with the stylographs on top. As you can see, we are looking for accuracy, but also simplicity and ease of visualization. So sometimes I'm not drawing all the details. In fact, I'm only drawing the details I am interested, which are the overall shape of the parasite, especially the hump, the spherical hump that appears on them, the position of the nucleus and how it moves, and the position of the malaria pigment, which is nothing more than a degradation product of digesting blood. And also the cytoplasm. The parasite it appears to have two types of cytoplasm. One is very granular, and the other one is like transparent. So I'm trying to portray all these details, and I'm doing that with a technique called stippling, which is similar to pointillism, but using only one color. Pointillism uses different colors to achieve color blending, but here we are just going to use one color, and this technique is called stippling. And you can achieve different effects accordingly to how spaced the dots are, and also the width of the stylograph. So then, because I am really not good at drawing, what I did was to scan them and then fix them a little bit in the computer. This is not cheating. Remember, we're not making art. So yeah, after fixing them a bit, this is the final product. So now we have the complete development of the malaria parasite within the mosquito. First, starting here, down here, in the left, we have the zygote, and the zygote differentiates into an ookinete within the blood bolus of the mosquito after a mosquito takes an infected blood meal. Within an hour, the zygote starts differentiating into this ookinete that migrates out of the blood bolus to invade the mosquito epithelium. So these drawings, this description was made by Janse and co-workers in 1985. And then this is what I described recently, the differentiation from ookinete to an oocyst. And remarkably, this differentiation process is quite similar to the differentiation process that happens in the liver. When the mosquito finally injects the sporozoites into the vertebrate host, for example us, then the sporozoite invades the hepatic cells and then it differentiates into a trophozoite. And it happens in a very similar way to what I have described. It happens during the differentiation process in the mosquito midgut.